first step is we're gonna take a beaker that's on the ultrasonicating machine, and we're gonna put our aluminum cones in at the bottom and make sure they lay flat. This way we just use less chemicals and it'll still cover the entire cone. And then we'll come over here. And what you want to make sure before we use the machine is we're going to check the machine to see if there's enough water at the bottom. So as you can see here, it's completely empty, so we have to fill that just with water from the sink is fine. So we'll just you just want to get it to a point where it's at least maybe like half an inch. You don't need to fill it up all the way because the beaker is so small. You don't want the beaker to be sliding around. So you can see here now, that's like about a good, I'd say maybe it's a quarter of the operating level. That's about where I like to put it. And that's just because you can see here, when I put the beaker in, it's still kind of bouncing around a little bit. So when I put the liquid in, it should be okay. So basically it's hitting about half of the height of the beaker, which is about what we want for the water. So the first step is we have to put a liquid into the cones. So in our case, we do it twice. So the first time we do it is with methanol. So you'll find it in this fire cabinet. So open that up and you'll see right here, there's methanol, which we need, and acetone, which we need. The order doesn't matter of the two chemicals. I just use acetone first because alphabetically, but that's just me. So there should be enough acetone to get this clean. Because basically all you want to do is just cover it. You don't want to overfill this thing because we're just wasting chemical. So we're good. It's just covering them completely. That's a little, like, maybe you could have done a little less. I just want to make sure this thing doesn't tip. So I'd say just like the water level, I want it around halfway. And then we'll place the beaker into the water. And we see it's not moving anymore, which is great. And we'll set it to 5 minutes. So it'll clean for 5 minutes. And what it does is basically just create water waves, and like these ultrasonic like water waves basically clean and strip the aluminum. And because it's such a heavy material compared to all our plastics, we have to do it for this material. So we'll set it for 5 minutes and we'll make sure to check on it so it didn't tip over. So now that those 5 minutes are done, we basically take a clean paper towel, lay it down, grab our beaker out of the water, and we have to take a pair of tweezers and just take the cones out, make sure they drain, and they'll dry really quickly because it's like an alcohol. And there's methanol and acetone, which are both dry super quick. So remember, we can't pour this down the sink, so we have a waste beaker over here that carries all of our waste. We'll just dump that in. And there's no need to clean this in between. We'll just put the methanol in. So the same thing as before. Make sure the cones are in first. Lay them flat. We'll add the methanol. Again, just enough to cover it. And weigh down. This one's a bit heavier, so be careful. Good. So about halfway again. And the same process as before. Just make sure, watch it for the first few couple seconds. Make sure it's not going to spin around. So now that all of our cones are clean, we can actually start mounting and cooking the bacteria basically. So I have a 1/5 dilution of pseudo here, and sometimes there can be bacterial clumps depending on how long it sits in the fridge. So when we take this out of the fridge, we want to shake it up for, I do 15 seconds. So I just make sure, this one has like a touch activation, so I'll just press it. There we go. So that was like 15 seconds. So now our first step is we have to pipette 100 microliters of this dilution onto this plate. This plate was cleaned basically by putting it in the bleach, soaking it for like 10 seconds, drying it off, and putting it in the deionized water to rinse off that bleach. So now that this piece is clean, we can take our micropipette and add a tip onto it, which might be a little difficult, but do not touch it with your fingers, just so we don't do any contamination. And then I'll adjust it at the base with my fingers now. So now we'll just make sure the setting. So the setting here says it's on 10 microliters. So when I hold it in my hand, a first stop is 10. Do not push all the way down when you're drawing up the liquid. Push all the way down when you're expelling it out. So I'll do 100 here. So I'll press down to the first stop, insert it into the tube, draw up the liquid. Now we have 10 microliters, and expel the whole thing out onto the plate. So I'll do that 9 more times. And make sure that you're trying to keep the liquid in the same area just so it doesn't spread out. 7, 8, 9, 10. So now I have 100 microliters on there. I will cap my dilution, put it to the side, and this is the eject button. So you'll push the button into the bio waste bag and it'll just get rid of the tip for you. So ideally at this point, you would have turned on your hot plate just to get it preheated. So we have to turn the hot plate on to 200, and also I set a timer for 2 minutes and 20 seconds. So you're gonna take the sample very carefully, and I take the large coarse tweezers, as well as a heating glove, which looks like this. So we'll go over to the hot plate. So as you can see, it's been at 200. I set my timer for 2 minutes and 20 seconds. I'm going to put the plate with the glass piece. Do not take the plate out. I'm gonna put that onto the hot plate and hit start on my timer, and we'll wait for that. So we just took the hot dish off of the hot plate and we put it onto this glove. You can wear the glove. I just like to hold the glove, but just be careful again, because it is like a pretty delicate when you're trying to move around the lab with it. So I have my tweezers again, because this thing is still very hot. I opened my hood beforehand, that way I can just slide this in and I put it on the steel because it kind of maybe singes this paper towel a little bit. So we're gonna wait for that to cool. In the meantime, we can start prepping everything else. So what we're gonna do is, first, we know what dilution we're doing, and we're gonna label 2 of these centrifuge tubes with the dilution and the date. And we're gonna label 1 Petri dish with the same thing. So for the format, I usually do bacteria then date. So for this one, it's pseudo, and the concentration is 1/5. And then the date is 10/02/2020. So I'm gonna repeat that 2 more times. And basically what's gonna go in here is the steel piece with the filter when we mount it and get ready to shoot. And these are gonna be for the inserts in the cones and also for just swabbing it off of the plate. These are a large pipette. And what we wanna do is make sure that we're adding 1 milliliter of liquid to one of these tubes. So when we swab off the bacteria, it will go into here. So to draw it up, remember not to get any air bubbles in it. I'm gonna clean my pipette first, which is always a good precaution. So I'm gonna just draw 1 mil into the beaker. So if you go pretty close to the bottom, you shouldn't get any air bubbles. And just draw it up slowly until I hit the 0 line, and that's 1 milliliter. And then I'm just gonna expel it into the waste. And always remember, anytime you drop anything into the pipette, do not put it back into the container it came from. So even if I had an air bubble and I wanted to draw something like bleach up, I would just have to throw that in the waste immediately. Now, just to get the excess bleach out of there, I'm gonna draw more air in and just expel it out. And that will just kind of knock some air around. Just try to get the bubbles off. So for example, let's say I had something that, like here, I had a huge air bubble like this. And I'm like, oh no, I need more liquid. So I'm gonna draw this out into here. That's completely incorrect. So just make sure anytime you draw something up, it has to go in the waste if you didn't get the correct measurement or there's an air bubble or something. So I'm gonna draw 1 mil of water now just to clean out the pipette of the bleach. So now I have a milliliter, and I'm just gonna expel that into the waste, knock the air around. So now this pipette is clean and I trust it now. So I'm going to put 1 milliliter of this deionized water for the samples. I just do this as a precaution so that anything that might have any bleach residue is going into the stuff that we use for cleaning, and anything that we're using bacteria, it has clean deionized water. So now I'm just gonna drop a milliliter of the clean deionized water for the samples, and I'm gonna put it into one of our centrifuge tubes. So now there's a mil of liquid into here. What we're gonna do now is that this thing is cold to the touch, so it's good to swab. The swabs I typically keep over the incubator. So we're gonna just grab 1 swab. They'll look like this. And take it into the hood. Peel off the outside wrapper about halfway. So I'm gonna take it, peel it. And you see here, there's ridges into the swab. So I want the shortest ridge. So I'm gonna try not to contaminate anything and break it off in the plastic so I can throw this in the garbage. And I can take the excess here and grab it with the end. So the tip is very clean. Throw that in the garbage. Now, again, before we pick off anything off of the plate, we have to get this a little bit wet. So we're gonna take our micropipette, put a new tip on it. So make sure anytime you're exchanging between bacteria and water to change out the pipette tip. And it's kind of difficult, but just make sure you're not touching it with your hands. So I got it on, and then I'll just push it down at the end, not near the tip. Make sure it's at 10 microliters again. We're just gonna need 10. So again, go to the first stop, draw up the liquid. Now I have 10 microliters in this, and try to pipette the 10 right in the middle of the tip. It might be difficult, but try to get it in the middle so nothing slides off. So you should form a nice little bubble like that right on top of this. Now what you want to do is be very gentle here and not be too drastic. Basically, I want to slowly tap the liquid into the tip of the swab. So tap it very gently. Don't shake anything around. We want the water all in there. So you can see it's kind of dispersing a little bit. Shaking it up. We just don't want the water running down the sides too much. And if it does run around the sides a little bit, that's okay. Just remember to swab with that side leading. So there's always water in contact and not just the swab. Okay. Okay, so now it's pretty much soaked in, and we're gonna take this swab, and we can kind of see the outline where the water evaporated and the bacteria resides. So I'm gonna sweep the perimeter and then the inside, and then I'm gonna put this into our water. So I'm gonna take the outside, swab it, see how it's already a little bit wet because of the water, and swab the inside like that, just to make sure I do that maybe twice. Swab the outside, swab the inside. Now you can see it's a little bit tinged. That's the bacteria, that's what we want. So I'm gonna put that tip down into the water. Now I'm gonna close the cap on my centrifuge tube. We'll put that to the side for now. So if we wanna make more samples, remember we have to take our plate and put it into the bleach first, let that soak, and then we'll dry it off and put it into the water just so it's clean for the next person to use. We're gonna take our swab with the water. We're gonna go to the vortex and do this for 15 seconds, which shakes off the bacteria from the swab and puts it into the water. We're taking 15 seconds, good. So now ideally all of our bacteria is in the water. And now what we do is we take off the cap from this, take our small pair of tweezers, be gentle here, try to grab the swab. So now that the swab is mostly out, I can grab it with my hand. So I'll grab it with my fingertips and just shake off any of the liquid on the sides of the centrifuge tube. Now that we're done with this, we have to put it in the bio-waste bag, and this is ready to use. So in the meantime, this is what our other tube is for. So here's the second centrifuge tube. This is gonna be for the parts in the filter. So we have to assemble this a certain way. Take our clean small tweezers, take a bottom piece, a top piece, and we need one of these aluminum cones. Always when you make these samples, remember to make them in two, or at least if you're making them in one, you have to repeat this setup for another one so that the centrifuge is balanced and so it doesn't explode. So basically now, this is where our filter is gonna be mounted. So we're gonna take a 0.5 microliter cut filter. So these filters are cut beforehand so they fit our dimensions exactly. So be very careful. Grab the two ends with your fingertips and pull it off very gently. It's kind of hard to do, but there we go. Because once they get knocked over, they're contaminated and it takes a very long time to cut the filters. So we want to keep those intact. So now I'm going to gently grab the white filter by the edge. I don't want to press anything in the middle because that's where we shoot. So I'm going to try to keep it as close as the perimeter as possible. So just kind of be very gentle, be patient. Remember there is a fan going, so these can be blown away from you. So please just remember that. I can see this filter kind of has a mark on it, so I'm not going to use that one. I'm going to throw it out. And these blue filters are just so the static gets away from the white ones. So just grab those. It doesn't matter if you grab those ones in the middle, just be careful. Remember what you're grabbing. That's going in garbage. And this one seems pretty clean, so I'm gonna grab this one by the edge. Now, be very careful again, try not to get too close to the fan. Hold the base down with your fingers and just place it on the bottom piece. Now, I take the edge of my tweezers and I knock it into place. So just making sure it's nice and centered. And when we screw these pieces in, it doesn't get crushed. So now it's nice and centered. I knocked it around my four corners. I know everything's lining up pretty nicely. It's good. Be very gentle again. These have threads, this has threads, so it's gonna screw into each other. Just be careful not to clip the edges. We want the filter intact. So I'm just gonna go in, try to be as straight as possible. Don't come in at any angles. Gonna go in, try to be straight as possible. Check it from the top. The filter is still good, and twist it in. And just be very careful because even at this point, the filter could get crushed in between. So just screw it nice and tight. perfect. And we can even do a double check. We can see that the filter is nice and flat on the inside. Then we're gonna take one of our aluminum cones, which have been cleaned, grab it, and just drop it in, and let it knock into place pretty nicely. There we go. We don't wanna push this too hard. Grab the end of the tweezer and just tap it gently at the four corners of maybe a couple times. No pressing or anything. We don't wanna cut the filter underneath. We just wanna make sure it creates a tight seal. So now this piece is gonna go into our second centrifuge. So it has a filter, it has everything it needs. What we wanna do now is take the bacteria sample, clean our pipette once more, just remembering to clean between each step. And it's okay if there's not air bubbles in this cleaning step, as long as you knock them out when you need to drop the liquid, that's okay. The exact measurements aren't needed when you're cleaning. We just wanna make sure the liquid gets through the pipette and just make sure it's clean. So I'm knocking out all the excess air. Now I'm gonna draw the 1 milliliter up. And sometimes we will do 1 milliliter or 0.8 milliliters. We'll let you know, or you know what you're doing, but it's somewhere in between depending on which cone you're using. So this aluminum cone can accommodate for the 1 milliliter, but if we're using the plastic cone, you can use 0.8 and it won't affect your results. So draw that now. We're gonna pipette that into the cone. This is a very crucial step. You don't want to eject the liquid very rapidly. You want to go nice and slow. What we want is a big bubble to form at the top, which you know is a good sign so that no water leaked out into the filter. We don't want that to happen yet. So I'm going to go really, really slow here. And basically I'm just filling up the cone really gently and making sure, we see it's already coming in at the top, and just making sure that it forms a nice bubble like that. And that's all you need to do. So then we're gonna put the lid on this really carefully again. Don't knock it around, and make sure you're always holding this thing upright. So I put it really gently. And now this is ready to go in the centrifuge. So basically we have 2 samples or a balance before we run this. So if I was making 2 samples, I would repeat this process maybe for a different bacteria. But if I just needed to make 1 sample, I would just add the aluminum cone and the 2 inserts. So the weight would be the same, and I would put that into another centrifuge tube. And then basically we'll close the lid, press the start button, wait for all the liquid to come out. When it's done, we'll check the inside to make sure there's no more liquid in the cone. And if it's good and it's clear, then go ahead. If it's not, just run it through again in the centrifuge. So if it's nice and dry, we'll take it into there. And what we do, I can show you now. So ideally, we would have this double-sided tape. So what my last label was for the actual piece of steel that we're gonna shoot the filter on. So I'm gonna take some double-sided tape that's about the width of this. Don't let it hang over. If it's smaller, that's okay. Just big enough so that it accommodates to the filter. So I'm gonna try this. Rip it off, and I'm gonna put it right on the steel. So let's just pretend I had the bacteria, it was done. I took the piece out, I carefully unscrewed it, and there was the filter. Let's pretend this is the filter. I would take the filter, mount it onto here, and just press the edges. Again, we don't want to touch the center, so just press the edges, and that's ready to be shot. So that's all you have to do. To clean it, so when you're done with this piece, remove the whole piece of double-sided tape, throw it in the bio waste, soak this in the bleach and then the water. Actually, sorry, these steel pieces that we shoot on, you can just put back over there. They don't need to be cleaned. We don't use them for anything. And any piece that's plastic like these is going to be soaked in the bleach, dried, and then soaked in the water. And these cones, you have to use the ultrasonicator to clean them. And anything like this, I'll show you really quickly. You draw up 1 milliliter of bleach, put it into the bacteria to. Press it in. We're gonna invert it. And then we're gonna put it for 15 seconds in the vortex. Now that that's 15 seconds done, remember to dispose of that into the waste. And we're gonna drop a milliliter of water for cleaning. Remember, we're not using the water for the samples. Making sure that's all knocked in there. Again, do the same thing, close it, invert it. 15 seconds in the vortex. And notice how the label's being kept on. This is just so I know that this thing hasn't been fully cleaned yet. So as soon as it has been, like right now, I will take this, dispose of it in the waste. Now that this centrifuge is nice and clean, try to knock out as much liquid as you can. It's okay if there's some in there. And then I can remove the tape and basically throw that tape in the garbage, put this centrifuge tube back into here, and now it's good for someone else to use. So anytime there's a tape or something, you know that that thing hasn't been cleaned properly yet, or someone's been using it. So that's just something for people in the lab to know. And that's it. That's the procedure.